Okay, in this video, we're going to continue the process of photosynthesis. This is part 2 of understanding how it is that plants can harness the sunlight energy and make food. Once again, here's a summary of the entire process that we're going to be talking about. We're going to start here with 6 molecules of carbon dioxide. 12 molecules of water are also coming into the process. Light energy is required along with some enzymes that the plant has. Remember, these enzymes are not going to be actually taking part in the reaction. They're assisting the reaction. They're catalyzing the reaction. We're going to end up with 1 molecule of glucose, C6H12O6, and 6 molecules of oxygen are released, along with 6 molecules of water. So there's a net of 6 water molecules going into the process. Now, we've already talked about the light-dependent reactions, or the light reactions. Now we're going to focus on the light-independent reactions, or the reactions that don't require light. They happen both in the daytime and at night, but they don't require light to happen. Now, at the end of the light reaction, we discussed that the sunlight energy had been captured into molecules of ATP and also this electron carrier NADPH. The next step in the process is to use that ATP and the electrons from NADPH to generate some glucose. This process is called the Calvin cycle, sometimes called the dark reaction, or again, the light-independent reaction. Now, just to let you know exactly where these different processes are occurring, here we have the plant leaf. If we take a section of the plant leaf and kind of blow it up here, we have some layers to the leaf. Most of the photosynthesis is taking place in these mesophyll cells. We have the palisade mesophyll, which are like in a squeezed together row up here, and then more spread out with some space in between them are the spongy mesophyll cells. But the mesophyll cells are the ones that are carrying out the photosynthesis. Then we have, I don't know if you've ever noticed, but leaves have little veins in them. You can kind of see them over here. And the vein itself is called the vascular bundle. And then the cells around there are called the bundle sheath cells. And these cells are going to become important in a minute, what we're going to talk about. Also notice that the leaf has this thing down here called the stoma, sometimes called the stomata, surrounded by these cells called guard cells. And this is actually a little pore on the underside of the leaf that opens and closes to allow the flow of gases in and out of the leaf. So if we take a close-up look at that stoma or stomata, when it opens up, carbon dioxide is allowed to flow in, and it makes its way into these mesophyll cells where glucose is produced. And water, along with oxygen, is released out of that same pore. Now, when we're looking just at the light-independent reaction, this part of photosynthesis is going to happen in the stroma of the chloroplast. We talked before about the thylakoid membrane, where all the light reactions are occurring. All the pigment molecules are located there and everything. Now we're talking about some processes that are going on in the fluid part of the chloroplast, on the inner part, where there's just fluid flowing around. The first thing that's going to happen is carbon dioxide gets bonded to a molecule of something called RUBP, ribulose biphosphate or ribulose bisphosphate. And it's going to be attached to this molecule of RUBP by an enzyme called RUBP carboxylase, and its nickname is Rubisco. So you'll hear people talking about Rubisco. That's the enzyme that's going to attach the carbon to the RUBP. This forms an unstable 6-carbon molecule, which breaks apart right away because it's unstable, and it breaks apart into 2 3-carbon molecules of PGA. This whole step from carbon dioxide being attached to getting to these 2 molecules of PGA is called carbon fixation. It's a way of the gaseous carbon being converted into a form that the cell can use. Right away, PGA is converted to PGAL. And for this to occur, the ATP is used. So the phosphate from ATP is added to PGA, and then a proton, a hydrogen ion, from NADPH. Both of these are attached to the PGA 3-carbon molecules, and you get this molecule called PGAL. Here's kind of an overview of that process. We have carbon dioxide being attached to RUBP with the help of Rubisco. You get this reaction intermediate, this unstable 6-carbon molecule, and it right away breaks apart into 2 molecules of PGA, or phosphoglyceric acid. And you can see kind of what that looks like. You're going to add on the phosphate groups, and you get PGAL. these two PGAL molecules are going to combine to form the glucose. So you've got two 3-carbon molecules with attached phosphate groups, and they are going to combine to form the glucose. The completed glucose has these phosphate groups attached, and what that does is it allows the glucose to be synthesized immediately into either sucrose, cellulose, or starch. So in a photosynthesizing leaf, you're not going to find a lot of glucose. Usually the glucose right away is going to be combined with other glucose molecules to either form sucrose, the large polysaccharide cellulose, or the large polysaccharide starch. That's the way that the cell's going to store up that glucose. Now also, along with this, some of the PGAL gets recycled to form more RUBP, because you have to keep the ball rolling, sort of. You can't just keep using up your RUBP. So some of it gets recycled back. The totals we end up with are 36 carbon dioxides to 12 PGAL. You get 1 glucose, and then 6 RUBPs get recycled back. Now, all of this we've been talking about has to do with these plants we call C3 plants, and it's a C3 plant because it's a plant that does photosynthesis using 3-carbon molecules. So we call it C3 carbon 3. But carbon dioxide and oxygen in these plants can move only at the stomata, and they're closed on hot, dry days. So these plants on a hot, dry day are going to reduce the amount of photosynthesis they're actually doing because they can't get carbon dioxide. They're going to close those stomata because that's going to limit water loss on a hot, dry day. They can't lose too much water, as you know. Now, when oxygen levels build up within the leaf, and it's a hot, dry day like that, some plants have this tendency to undergo this process called photorespiration. It's not necessarily the best thing for them to be doing, but it's just there. It's what plants will do. And it's because RUBP will bind to oxygen instead of carbon dioxide in this case, if oxygen levels are high. So some of that RUBP will be bound to oxygen instead of binding to CO2. Later, it can be converted back to CO2 to release that CO2 to make glucose, but it kind of slows down the whole process and makes it inefficient. So best for plants not to undergo this photorespiration. Another pathway, which is interesting, is plants that we call C4 plants. They use an intermediate instead of PGA or PGAL. They use a different intermediate, and it's a 4-carbon intermediate. And these plants, the way that they do photosynthesis allows them to maintain high photosynthetic rates even when it's hot and dry. In these plants, carbon dioxide is converted, instead of PGA, it's converted into something called PEP, and then eventually converted into oxaloacetate, which is the 4-carbon molecule. And this takes place in the mesophyll of the leaf. So in the place where normally this would happen, CO2 gets converted to PEP and then into oxaloacetate. Then the oxaloacetate is moved to the bundle sheath cells through the plasmodesmata, which you know what those are now, plasmodesmata. The oxaloacetate is moved into the bundle sheath cells, far away from where the carbon dioxide and oxygen are being moved into and out of the cell, so into the inner part near the vein of the leaf. The oxaloacetate gets converted back into carbon dioxide, and then the Calvin cycle can proceed. So in this case, the plants can close their stomata. They don't have a problem with oxygen building up and photorespiration occurring. Instead, they move the oxaloacetate into the bundle sheath cells away from where the oxygen is building up, and they can continue to run the Calvin cycle that way. Ingenious. Now, these are the type of plants that are C4 plants. There are grains and corns. Sorghum, I don't know if you've heard of that. Maize or corn. Sugarcane is a good example. And then millet, which is a type of grain. So C4 plants are the kinds of grains and corns and things like that. Very efficient at photosynthesizing. Here's an overview of this process. So in this process, we have the mesophyll cells here. CO2 comes in. It's converted to PEP and eventually to oxaloacetate. The oxaloacetate is moved into the bundle sheath where it's converted back into CO2, undergoes conversion into, first 3-PGA and then into glucose, and the products are sent out the vein to the rest of the plant. So understand that the Calvin-Benson cycle, or the Calvin cycle, the dark reaction is happening over here in the bundle sheath cells instead of in the mesophyll where it would occur in a C3 plant. So just comparing these two processes, we have C3 plants here and C4 plants here. In a C3 plant, we have our mesophyll cell, CO2 comes in, ribisco is our enzyme that's going to help us to convert it into glucose in the Calvin cycle. In C4 plants, we have CO2 coming in the mesophyll cell, and PEP, the enzyme here is PEP carboxylase. It's converted into this C4 intermediate. It's the oxaloacetate. It's going to be transferred to the bundle sheath, where again ribisco is involved in converting glucose, in converting the CO2 into glucose in the Calvin cycle. So there's an extra step here, and the Calvin cycle is actually taking place in the bundle sheath. There's one more process that different kinds of plants perform, and these are CAM plants. CAM stands for crassulacean acid metabolism, if I said that right. This is another way that the plant, another different kind of plant, and a different kind of form of photosynthesis altogether. These plants open their stomata only at night. So they live in extremely dry conditions, and they grow very slowly because of this. So they're only getting a little energy. They have to grow slow. But they have an adaptation for an extremely dry environment by only opening their stomata at night when evaporation would be less. It would be cooler and not so much sunlight. So what they do is they fix carbon into another different intermediate molecule at night, and then the Calvin cycle runs during the day. So it's kind of different from the way C3 plants do it. Here are a couple of examples of CAM plants. These are both succulents. I'm not sure exactly what this one's called, but you could think of like your aloe vera plants. This is a jade plant. Those plants that have a lot of water stored up in their leaves. I believe cactus are also CAM plants. And here's an overview of the comparison for the C4 and the CAM pathway. Notice it says that in the C4 pathway, such as that with sugarcane, there is a spatial separation of steps. So you're separating the light reaction in the mesophyll cell from the Calvin cycle in the bundle sheath cell. So you have a spatial separation. You're separating these two processes. In CAM plants, they have a temporal separation of steps. That is, rather than separating them physically, they separate them in time. So in this case, they're doing the carbon fixation at night, and in the day they do the Calvin cycle. So you can compare those two. And a final comparison looking at C3 plant versus C4 plant versus CAM plant. We've looked at this already. In a C3 plant, we have RUBP, PGA converted into PGAL and eventually into glucose. In C4 plants, we have a separation of the steps, the light reaction here, carbon fixation, and then the Calvin cycle here in the bundle sheath cell. And then in a CAM plant, we have this C4 intermediate being produced at night when the carbon dioxide is available because the stomata are open, and then the intermediate being converted back into CO2 for the Calvin cycle to run during the day. So that's it for photosynthesis, and we will see you later.